Right on. Welcome to the Brassica Crops IPM production workshop. And we'll be going over some introductions, talking about production and scouting techniques, followed by entomology, and then disorders and pathology. I'm Travis Cranmer, vegetable crop specialist for alliums, above ground, leafy and head brassicas, as well as leafy greens, spinach, lettuce and celery. And Dennis Van Dyke, who is also presenting today. Dennis, are you there?
I'm here.
Right on. Want to introduce yourself?
Sure. Also vegetable specialist with OMAFRA, but I cover potatoes and root vegetables. So that would include the root brassicas, rutabaga, radish, turnip, horseradish, that kind of thing.
Right on. So we're recording this session. If possible, please stay on mute during the presentation so speakers can be heard clearly by all the participants. Disable your web camera if doesn't need to be used, especially if your internet connection is poor. And feel free to use the chat function for questions on Zoom or Slido, and avoid sharing links in the chat. So here are our vegetable crop specialists with OMAFRA. We have Elaine Roddy and Amanda Tracy based out of Ridgetown, and myself and Dennis based out of Guelph, and the representative crops that they look after. So coming up next week, we have the carrot and onion workshop on Thursday, the potato workshop on the following Tuesday, followed by sweet corn, peas and beans Friday, and cucurbits on May 26th. Once these workshops have been recorded and we've had a chance to look them over, they'll be posted on the On Hort Crops YouTube channel. Right on. So we'll get started with production and scouting techniques. So the Brassicaceae family is very diverse and includes all of the vegetables listed on the right, and even some that aren't even listed, like horseradish. And in Ontario, I would say cabbage covers the largest harvested area with over 5,000 acres, followed by broccoli, rutabaga, cauliflower, and Brussels sprouts. Over the last 10 years or so, the average price per pound in cents has been increasing for most of the crops, especially broccoli. And production takes place across the province. However, when it is broken down by county, these are the counties where the highest acreage has been grown in the past for your head brassicas. And then for leafy brassicas, these are the main growing regions where you could expect to find these leafy brassica greens grown. So brassicas are super healthy. And so naturally, these are great to be consuming in large doses, and they're awesome. It is thought that brassicas have been cultivated as crops for thousands of years, and over time it has been bred into a wide variety of cultivars, with specific traits being selected for. So in kohlrabi, a large stem was selected for, the leaves were selected for in kale, lateral leaf buds in Brussels sprouts, and flower buds and stem in cauliflower and broccoli, and then roots were selected for rutabaga or turnip. With such a great diversity in cultivars, there are a bunch of ways to classify brassica crops. We could classify them as if they are short or long season. Many are grown for early, mid, and late markets, especially cabbage. And then there are quality differences among types. So early cabbage is the first to get to the market, but it has a short storage life. So let's start with cabbage. Most of the head brassicas are biennials, grown annually. They're great for different markets. There's early, mid-season, and late. So your early is primarily your fresh cabbage. Your mid-season includes those fresh coleslaw cultivars, and then the late cabbage is for storage, where you can keep pulling that cabbage if you put something quality in for many months after it's been put into storage. Can also be classified by what color the cabbage is or the head shape, with your Chinese varieties generally being the pointed cultivars. Round head is your typical varieties, and those with the flat head are your kraut varieties. It's annual grown for florets. There are 3 types of broccoli: salabrise and sprouting, or broccolini. And these are either grown for your main head, for bunches of heads, or florets because the head doesn't develop fully. Next, we have cauliflower. Once again, there's early, mid, and late markets, with the late type being used for processing. There are a variety of colors available, but obviously white is the most common. And keeping that head white, these are tied when they reach 8 to 10 centimeters in diameter. Brussels sprouts is my personal favorite. You have those lateral buds along the stem, and it is topped when it reaches a certain height, and that promotes growth for the buds that still exist along the stem. With kale, it's a very high in vitamin K, a high value of other vitamins, can mature in as little as 55 days from transplanting or 70 days from seed. There are 4 different types of kale: curly, Jacinto, Siberian, and red borer. And they're grown for 2 different markets, with most of the kale grown in Ontario for the fresh market. Radishes, there are so many different types of radishes. They're mostly short season and can mature in as little as 4 weeks given good growing conditions. Often grown for the fresh market, but can be stored for small amounts of time and sold bagged or bunched. Turnip and rutabaga. So turnips are normally white or white and purple, while rutabagas are purple and sometimes yellowish, depending on the cultivar. Rutabagas are often grown larger than turnips, and they stay tender at these larger sizes. With leafy brassicas, there are so many different types. They vary in leaf size, texture, taste, and growth habits. Some are even enjoyed by cows. And I would say, in general, these brassicas take a shorter time to reach maturity, and many can be grown in high tunnels throughout the fall, spring, and even winter. And obviously their growth rates decrease at these lower temperatures, but they're still harvestable. For seeding and transplanting, your planting style depends on the cultivar brassica. So radish, turnip, rutabaga, and ethnic brassicas, leafy brassicas are generally planted by seed, while your head brassicas and sometimes kale are transplanted. Brassicas can tolerate a wide variety of soil types. In general, your early seeded crops are grown on sand, and I would say heavier soils are better for mid to late season brassicas, especially if they are fully saturated in the spring and it's hard to get into the field. Short season leafy brassicas grow well on high organic muck soils. As far as drainage goes, well-drained soils are favorable for most crops, but they don't hold water well and often require irrigation. And brassicas prefer soils with pHs between 6 and 8. As far as fertility goes, recommendations vary depending on the crop and soil type. Brassicas are pretty heavy consumers of nitrogen. Low nitrogen can reduce quality. I've included these fertility tables to have in the slides to be viewed at your own time. And this course is, our workshop is designed more for field scouts that are identifying pests in the field. As far as plant spacing goes, it's listed here as well. Radish is probably the one that's planted closest together. And depending on the head brassica, you could go as far as 3 feet apart between plants. So if you're planting on sandier soils, they should be irrigated to get a good yield every year, but not too much water because that favors pathogen spread and some nutrient disorders. Once the crop is harvested, it is essential that there is as little bruising as possible to prolong the storage life. It's also important to remove field heat rapidly as possible because you know, if the crop gets harvested and is left in full sun for several hours, it's not going to store as long as if it can go right into the shade or cool down. In general, these brassicas store well at temperatures just above freezing at a relative humidity of around 95 to 100%. Moving on to scouting. So why scout? Scouting is the foundation of all IPM programs. If you manage to join the Intro to IPM Scout Training Workshop, they covered this in detail, and I'm just going to cover the key points. The basis of IPM is an approach to pest control that considers all management options to maintain pests below an economic injury level. So it's not just counting how many pests are on the leaf, but we also want to understand the crop, the cultivar that's being grown, the production practices at which it's being grown. We want to understand the biology of the pest and know when the right time to apply a management strategy is. And we want to be able to apply that management strategy on the pest that is actually in the field. So correct identification is essential. So before going in the field or as you go in the field, review those scouting calendars. I'll be showing the Brassica one shortly. Learn to identify all life stages and damage of the various pests. Scouting once a week is ideal, but twice is preferred. Keep a field map and mark on it on a weekly basis. And that way you can identify patterns or be more likely to return to a problematic area in future weeks. In each field visit, record the date, stage of crop development, the disease severity that you're seeing, the level of insects and the amount of damage that they've caused and what leaves that damage is on. If there have been any pesticide applied or any other control methods. So heading out into the field, it's really good to make a tote with a whole bunch of varying amounts of equipment. Do it now because you won't have time later in the season. And here is a list of tools that make monitoring easy. I'd like to highlight the trowel. That's really important for identifying pathogens on roots, and collection bags and vials. It is so handy to have a box of Ziploc bags, and that way if you find something that looks like a virus or you don't know what it is, you can get it in the bag, seal it, get it out of the field without infecting things as you exit the field. So here's that pest activity calendar that I was referring to before. And so we have pathogens on the top and insects on the bottom. For going out into the field, you want to scout as many different sites as possible. So choose sites that differ as much as possible when it comes to maybe a high spot in the field or a low spot where flooding occurred early in the season. Maybe a spot that's close to the greenhouse because these microclimates might be a refuge for overwintering Brassica pests. Good spot to check is along the sprayer row, but also far away from the sprayer row, as well as the field border or the field entrance. So identify the block that you're scouting, choose a pattern, whether it's a V or a W or an X, and if it's a W, you're basically choosing a point at each of the different points of that W, or at the X, it's the points in the middle. In the V, it would be 1, 2, 3, 4, 5. At each of those 5 spots, examine 5 plants, look at every leaf of the plant, look at the stem, examine the crown, look for anything that looks abnormal, and be as detailed as possible with each of these 5 plants. And so counting these insect pests or things that you're finding in the field allows us to determine if we are above or below a given threshold and whether it is economically worth managing the pest. There are some insects that can be left in low numbers throughout the growing season without compromising the overall crop quality. So in brassicas, as far as insect traps go, they're not generally used, and I think the exception would be swede midge. There are pheromone traps available for swede midge. Most of the time, we're doing insect counts on plants for your butterflies or your moths, like imported cabbage worm, diamondback moth, cabbage looper, or flea beetles. Temperature and leaf wetness data aren't generally used in brassica crops. It's usually a presence or absence of a given pest or pathogen. There are degree day models for cabbage maggot that have been developed, and it's reported weekly on the On Vegetables blog in the vegetable crop report for 9 regions across the province. So if there are no thresholds, the grower must make decisions on when to apply the pest control product. And in most cases, especially with fungi, no lesions are acceptable on the crop. As a scout or a consultant, personal exposure to these products is up to you. You are in control of your exposure, the amount of time that you spend in the field, and the personal protective equipment that you're wearing. So communication with the farm manager, the grower, is key. Learn to recognize signs of spraying, whether it's sprayer row tracks, the residues on the leaves, or if there's that a distinct odor in the air. Do not enter the field until you have determined it is safe to enter. Follow that re-entry interval on the label, and labels are available through the PMRA label search, and summaries of those labels are found on the vegetable crop protection guide, as seen on the right of this slide. So PMRA label search can be, if you type in PMRA label search to Google, this webpage should come up as your first hit, and enter the label title and click on the number on the left-hand side, and you will get a full PDF of the label. If you're looking for a summary of what's registered for various vegetables, the Vegetable Protection Guide 838 was just published and is available for download at the link at the bottom right-hand corner. If you're applying products, ensure that you have a pesticide license. The Ontario Pesticide Education Program is currently booking courses online and can be reached by going to www.opep.ca. And when you're visiting multiple fields in a day, practicing good biosecurity is key. Soil-borne pathogens can easily move between fields on boots, tires, equipment, and in brassicas, clubroot is a very destructive soil-borne pest that we will talk about in the pathology section. And the main method of spread for this pathogen is through dirty equipment and tires. So keep that in mind. If your vehicle is dirty, park it on the road and walk into the field. Clean your boots as you after you've entered a field before you go into another field. And if you're unsure of any pathogens, send samples for confirmation to know what you're dealing with. And with that, I'd like to thank the previous authors of this presentation on this slide. And you can enter any questions you have into the chat or email us. And we'll take a quick 10-minute break and start the entomology section when we return. All right, thanks for filling those questions out. We'll start with Brassica entomology next.
Sounds good. Thanks, Travis. I will take over. So happy to go through some of the insects you're going to see when you're scouting for brassica crops this year. If you have any questions, either type them into the chat, raise your hand, pipe up, put them in Slido, whatever you feel most comfortable doing. Just at any point, get my attention if you have a question. So first thing I want to talk about a little bit is the insect pressure at different development stages. So the stage at what the crop is at is important in what you're scouting for and what's important in terms of insects that you'll be scouting for. So at the early season seedling stage or recently after transplanting stage for some crops, in general, the plants are very susceptible. They're very susceptible to a specific number of insect pests as well. So in particular, you see that list there: maggot flies, cutworms, imported cabbage worm, flea beetles, wireworms, snails, and slugs are all sort of early season pests that can do a lot of damage. So a lot of these pests, for example, will attack the stem. They can kill the stem and basically kill the plant right away. So it's important to do a good job of scouting for those specific pests at that growth stage of the crop. At intermediate growth stages, most brassicas can tolerate a little bit of damage, especially if it's sort of leaf feeding insects, except for leafy vegetables that are selling that leaf part. They just have a much lower tolerance, but in general, those head brassicas or root brassicas can tolerate some damage. That's not necessarily the case when you have some brassicas like broccoli, like cabbage, where as those leaves start to cup and you start to create a head, you need to make sure that there's no insects inside there trapped and can do a bunch of damage where you can't see them. So some of those pests, you might be looking for the sweet midge or your lepidopteran pests. So you're basically caterpillar feeding pests. And then also later on in the season at head formation, there's very little damage that is tolerated. There's very little frass that's tolerated when you're shipping, a cabbage to the producer. They don't want to see either caterpillars or ragged holes or insect dung inside their nice cabbage. So generally that's frowned upon. So those sort of later season pests that you'll be looking out for are those lepidopteran pests, so your caterpillars, thrips, aphids, and sweet midge. So I'm going to go through some of the early season pests. So I'll start first with our Delia maggot flies. So The Delia species are a wide species of flies. Their maggots do quite a bit of damage. You can see here, these are three of the probably the most common ones that you'll see in Ontario is seed corn maggot, onion maggot, and cabbage maggot. Those are also the three that probably do the most damage, especially to horticultural crops. You can see by their pins, they are sort of fairly similar. Seed corn maggot and cabbage maggot are fairly similar size. They all sort of have gray to brown bodies and reddish eyes. So it can be quite difficult to sort of tell the difference between a lot of them, unless you're an entomologist, for example. But I'll get into that in the next couple of slides as well. But in general, all these flies are very similar, sort of from the same family, and they all do sort of similar damage. I'll start with the most important one for brassica crops is the cabbage maggot fly. I mentioned before, it's gray body and sort of red eyes. You can see here, sort of grayish body with the red eyes, about half the size of a house fly. If you see them on the crop, or if you see them on your sticky traps, they can infect all brassicas. So any brassica crop that you'll be scouting from canola to cabbage can all be infected by cabbage maggot. They overwinter here in Ontario. So these adult flies that have overwintered as pupae will emerge in the spring. So you'll have one flush in the spring of adults emerging from the soil. The adults will mate. The female will lay eggs at the soil near the base of your brassica crop. So any of the brassica crops you'll see the eggs can be laid there at the base of the soil. Those eggs are oval, white. They're quite small, only a 1 millimeter long. Sometimes you can see them if you're really doing your spot checks. You can notice them, but they are sort of hard to tell. And those pupae are tan to brown. You're not really going to see them unless you're sort of digging up the soil. So those eggs will then hatch, depending on the temperature, between 2 to 10 days, so quite quickly, especially if it's warmer. They can grow up to 7 millimeters long. They sort of have a little pointed head and a blunt rear end. So you can see here sort of a blunt end and a pointed head with sort of a black, almost feeding tooth you can see sometimes. They'll feed on the roots, they'll dig their way in a tunnel, and they'll feed, do a bunch of damage. You can see here on the stem and on the roots, basically at the base of the plant. They will then pupate, so fall into the soil. They'll go through their brown pupate state, and then they will develop into a fly and then emerge 2 to 3 weeks later, also depending on the temperature. So typically we see 2 generations in most of Ontario. We do have a degree day model that's been established through research back in the 80s, 90s that we do use. We do report on, as Travis mentioned, weekly on vegetable crop report. We'll have an update of the different regions and whether or not you'd expect to see cabbage maggot in full peak or full flight. So it's basically a base 6 degrees Celsius degree day model, and it sort of predicts when you'll first see adults, when your first peak will be of those adults, and then later on through development where your second and potentially your third peak. Some southwestern Ontario areas can sometimes have a third wave of cabbage maggot as well. So I mentioned there's other Delia species within that complex. Seed corn maggot also has the potential to infect brassica crops. Onion maggot does not. Onion maggot is only a pest of allium crops, but seed corn maggot for sure can infect brassica crops. In order to tell the difference between seed corn and cabbage maggot can be quite difficult to next to impossible, unless you're an entomologist and you have your own dissecting scope at home. But basically you need to count, the number and location of the leg hairs in order to tell the difference between the 2 species. In general, they're basically the same size, sort of half the size of a house fly. They have gray bodies and reddish eyes. The difference, I guess, in host range is that the seed corn maggot has a very wide host range. So it's sort of corn. If you scout in corn, you may be familiar with seed corn maggot for sure. But it's also attracted to dead or decaying matter. So if you have, a cover crop or something that's been terminated, a lot of times it'll attract it. Also attracted to fields that have a high amount of organic matter. But sort of similar life cycle. They overwinter, but they have a much quicker turnaround. So seacorn maggot have at least 3 peaks usually during the growing season, and a much tighter generations basically than cabbage maggot. Cabbage maggot takes quite a while for between generations to emerge and to develop, whereas seacorn maggot has a very tight generation turnaround. You can also tell the difference if you have maggots between what sort of the tapered ends look like. Again, not overly helpful unless you're an entomologist and you have a dissecting scope at home, but if you do have major problems in your field that you're having and you're not too sure what the pest is, we'd be happy to take samples and we can identify sort of the species from there. It is quite difficult just by looking at them. So moving on to another early season pest, wireworms. Wireworms are actually the larvae stages of the adult click beetle. These wireworm larval stages can stay in the ground for many years, 3 to 6, depending on the species. They also prefer sort of high organic matter soils. They enjoy monocot crops, especially grass crops or grain crops. So if you had a crop that was in pasture or sod or has a lot of grains in the rotation, generally your wireworm pressure will be higher. Usually you see the damage earlier on in the season, and that'll be attacking sort of the roots and the basal plate that you may see damage. To scout for these or to monitor for them, often we'll use bait stations. So there's sort of 2 options. One, you could basically just plant a carrot in the ground. They'll be attracted to the carrot and you'll see the actual feeding damage you can see here, very visual, very obvious. Another way that you can scout for wireworms is to use another bait. So generally like a bag of corn or something like that. You dig a small hole, cover that with a little bit of, well, basically clear plastic, so it'll warm up and that will attract the wireworms to there, and you'll also see feeding damage in there and will attract the larval wireworms to that area. So we typically use 4 traps an acre. A lot of traps recommended. If you are doing any neonic scouting for field crops, you're probably well aware of wireworm scouting, and you can basically use the same techniques to scout in brassica crops as well. So moving on to probably one of the most important complexes in brassica crop scouting, the looper complex, or the lepidopteran pests, basically your caterpillar pests. This is, I'm not sure who took this photo, but this is sort of an invaluable photo. I tip their hat to whoever got that all in one picture. But you can see here the 3 main lepidopteran pests that you'll be looking for in brassica crops: diamondback moth, imported cabbage worm, and cabbage looper. So diamondback moth, DM, we kind of call it; imported cabbage worm, ICM; and cabbage looper, CL. You can see here their caterpillar stages all sort of look a little bit similar, right? Sort of a lime to green caterpillar. The diamondback moth and cabbage looper are more moss, so they look a little bit different, whereas the imported cabbage worm is more of a white butterfly. But you can see here some of all the different stages of these 3 main pests. So let's start talking about diamondback moth first. So those are those eggs you see here, sort of that cluster of greenish to clear eggs in a bunch. These do not overwinter in Ontario. Only imported cabbage worm really regularly overwinters in Ontario, but diamondback moth usually comes up from the south. So that'll be important a little bit later when we talk to when you see them. That there is I guess a certain exceptions. If you have cull piles that have large infections from previous years, you will see diamondback moth overwinter, but that I would say is rare, and you should always be scouting cull piles if there are any around where you're scouting. But as I mentioned, eggs are kind of scale-like. They're laid in small groups. And the larvae here, you can see they are sort of hairless, they're sort of smooth, and they're feeding. When they feed, they create sort of windows. So what they're doing is they're eating the leaf tissue and leaf material and the photosynthetic area, but they're leaving that waxy cuticle layer of the leaf on. So what that does is create sort of like a window appearance where you can basically look through the leaf, but you still see that waxy cuticle layer. So it almost looks like a window pane effect. So that is diamondback moth that feeds like that, where it doesn't eat completely through the leaf, just up until the waxy cuticle. They can cause some pretty severe economic damage, especially at certain times, you know, when the cabbage is starting to form a head, or if it starts feeding on the growing point of the plant, it can do some severe damage. If you see and look on the underside of leaves and you sort of see this webbing, this is the pupae. So what's typical about diamondback moth is it has sort of a gauze-like cocoon. So a very distinct sort of cocoon layer. You can see almost looks like gauze, and that'll be on the underside of the leaf. You may recognize this from earlier, but that'll be distinct. So green larvae feeding damage looks like windows, so they don't eat all the way through. There's no hairs on the larvae, and you flip underneath and you see sort of this gauze-like webbing cocoon. You may see some adults flying around, not so likely. These are usually nocturnal, but if you do see any adults flying around, the diamondback moth sort of has a distinct pattern on its back at rest. So you can see here, it sort of has diamond shapes almost on its back, hence the name. So if you do see a small, they're quite small. If you do see a small moth hanging around a cabbage field and you're seeing damage or larvae like that, then look for that distinct diamond shapes on the back. So imported cabbage worm, so that middle picture, right, that sort of yellow bullet egg is imported cabbage worm. This does overwinter in Ontario. We do have local populations. The eggs are yellow and sort of ridged, almost like a bullet, very distinct. The larvae are quite mobile, but they are quite sluggish. They're I would say a little, usually large, plump, and the distinct ones about these are they have short hairs on them. They're sort of fuzzy, almost, I would describe them. So sort of plump and fuzzy. They leave lots of frass. They do not leave windows. They eat right through the leaf, and they'll create sort of large ragged holes. And the distinct one about this one is this is the one you'll typically see. If you hear a grower talking about, oh, the white butterflies are out, that's imported cabbage worm. And you will see these white butterflies flying through a Brassica field during the day as you're scouting during the day. So that's distinct about them. So those yellow bullet eggs underneath the leaves, a bunch of ragged holes that ate all the way through, very plump and fuzzy larvae, and then white butterflies flying around. That's your imported cabbage worm. Moving on to cabbage looper. You may also recognize one of these pictures. This one does not overwinter in Ontario either, just like diamondback moth. These are the big round white eggs that you saw on that first picture, usually found sort of in a couple, in a group. So there'll be a couple of those little white eggs on the other side of the leaves. The larvae are inchworms. So they are green and they don't have hairs, but they are distinct in the way they move around as that they're an inchworm. So they'll move in that distinct, you know, curve back, move the back up, go forward, inchworm style. They also leave green frass usually in piles. So the imported cabbage worm, the frass will be all over the place. Cabbage looper, they'll be in piles. They also do not leave windows. Only diamondback moth will leave windows in their feeding. This also creates large ragged holes, just like imported cabbage worm does. The cabbage looper adult is also a moth, kind of modeled. It does not have that distinct sort of pattern on the back, just like diamondback moth does. 3-centimeter wingspan. You likely won't see a lot of these, but if you do notice a moth in a cabbage field, it's likely that it's a cabbage looper. So all these lepidopteran pests are important. It's important you know all of them, but they also vary in their ability to feed and their ability to do damage. So when we are reporting on these caterpillars, it's sort of important to give some context of relative damage or relative risks. And to do that, we use a cabbage looper equivalent to sort of even the playing field. So the cabbage looper equivalent, right? So the number of cabbage loopers that you see for cabbage looper is 1. Imported cabbage worm, it's half. So basically 2 imported cabbage worm do the same damage as 1 cabbage looper. That's sort of how that works. And then 0.2 for diamondback moths. So about 5 diamondback moths do the same damage as 1 cabbage looper. So how does this work in an actual scouting situation? Well, let's say you have your notebook, you're doing, so all of sort of the counts and things that we're going to base off of, we base it off of 25 plants. I would say that is just about the minimum you'd ever want to scout in a field. So going specifically to 25 plants in different spots and really counting the things I would, if you're a scout in a larger field, 100 is pretty reasonable. But for the sake of argument here, we're going to use 25 as our example. So we've gone through our field. We have counted the different types of loopers and the caterpillars that we saw. We've identified them, and we've come up with 10 diamondback moths in the field, 4 imported cabbage worm, and 9 cabbage loopers across the field. So we use our cabbage looper equivalent, right? So multiply it by 1, 0.5, 0.2, and end up with 2 diamondback moths, 2 imported cabbage worm, and 9 cabbage looper in terms of cabbage looper equivalent. So what we're going to do, add those together, divided by the number of plants, so you get a per plant number, and end up with 0.52. So what does that number mean? I'll tell you. So that number then relates back to our threshold numbers. So this is the important ones here. I wonder if I have something. No, not going to work. Okay, I'll leave it. So your threshold here for cabbage is 0.3. And a cauliflower broccoli, it's 0.2 to 0.3. So what was our number, if you remember from the previous slide? 0.52. That puts us over the threshold for our lepidopteran pests in this case. So in general, whether you're using 25 or 100 plants, count the number of the different ones, multiply it by its cabbage looper equivalent, add them all up, divided by the number of plants that you scouted, 25, 100, and get your compare it to your threshold here. A lot of times you can have scouting reports that'll have the thresholds on there already, and it'll make it easier for you to do these calculations. If you, come on. There you go. If you don't want to worry about cabbage looper equivalents, there's also an easier way to do it, and just do percent infestation. So in this example, if we have 25 plants, right, we're basically just adding up the number of plants that you see that have at least 1 caterpillar on them, regardless of whether it's diamondback moth, imported cabbage worm, or cabbage looper. Multiply it by 4, and basically our thresholds here are 20 to 30% before the head is filling. So early on, you have a much higher threshold, much lower threshold after the head is filled, right? Less tolerance for damage once you start actually making that marketable head. Cauliflower and broccoli, same before the head starts to be produced. You have the same sort of 20 to 30% can tolerate the damage, but once that head starts to fill, even lower threshold for damage, 5 to 10%, really low tolerance. So a little bit easier than the cabbage looper equivalent, you don't need to worry, but also a bit less accurate in terms of what the risk to the grower is and the risk for damage. You may see other caterpillars within a brassica field as well. These are generally not as important. Good example is a zebra caterpillar, or this is an armyworm. It's a type of armyworm. There's also other armyworms that you might see. Generally, with these zebra caterpillars and other armyworms, what you'll see is 1, 2, maybe 3 plants in one spot totally infested and totally defoliated by these armyworms, and you won't see it in the rest of the field. So they can be quite sporadic. do not include these counts in your main caterpillar counts. A lot of times it's not economically worth it for a grower to spray an insecticide for those 2 or 3 plants, but just make sure that you're not missing larger infestations across the whole field. Cutworms are another important early season pest. They are most damaging early in the season, especially when it's recently transplanted or if there's young seedlings. What they will do is they'll hang out in the soil and they will emerge at night. They are night feeders. They will find the stem, they will feed on the stem and basically cut the stem right off. It almost looked like somebody came by with a pair of scissors and snipped those stems right off, or you can see it almost looks like a beaver feeding damage on a tree trunk. So you'll see in an area, maybe a 1, 2, a line of 5 of all these seedlings just totally being fed. If you dig around that area, you'll likely find one of these cutworm larvae in the soil in the area. So typically they're coming in from the area, from the border areas. If you do see this sort of damage, you'll likely find one of these cutworms in the area. There's no real thresholds. It has a very wide host range. If you're seeing it in brassica fields, you're likely also seeing it in other horticultural fields in the area. These will sort of flush in to the area and you'll see sort of a large amount in 2 weeks. So just be mindful. Subscribe to our vegetable crop report. We'll talk about cabbage or cutworm pressure if we're seeing damage, and that should give you a good indication of whether or not they have flown into the area. So let's get into aphids. So more of a mid-season to late season pest. There's several brassica crops and several species that'll target brassica crops. So your green peach aphid, I'm sure you may have heard of, very wide host range, definitely feeds on brassica crops. There's the cabbage aphid, the turnip aphid, and the turnip root aphid are also very common species that you'll find in brassicas. You'll find them underneath the leaves. So make sure that when you're doing your spot checks, you're looking underneath the leaves, looking over the whole plant, looking at the stem, and looking under the leaves, and you'll find typically clumps of aphids. Very high reproduction, especially in hot or warm temperatures. These populations can explode within a week. So ensuring that you're really checking and looking for any sign of aphids underneath the leaves is really important, because by the time you come back in the next couple of days or a week or 2 weeks from now, then they could be sort of out of control. Not only do they feed and suck on plant juices, but they also transmit plant viruses. So in this case, turnip mosaic virus is one of the most common ones that we're worried about. So the feeding can sort of stunt and distort, but the virus also stunts and distorts to tissue. Aphids will produce honeydew. You'll see sort of a sooty mold, sort of a gross thing on the underside of leaves. It can also secrete toxin and leads to leaf curling. So you'll see sort of distorted and weird looking leaves if you have really high infestation of aphids on the underside of the leaf. When you're monitoring for them, do it when you're doing your spot checks, mostly checking the underside of the leaves. They will all be different colors. Even the green peach aphid can range from green to pink to a whole bunch of different colors. So color doesn't necessarily distinctive for these different species, but turnip aphid you see here, cabbage aphids are usually more darker, from almost like a purplish. The species doesn't necessarily matter as much, but you can get them identified if you'd like. Note the distribution. Relay back to the grower whether you're seeing it in 1 or 2 brassica crops or plants, sorry. Or if you're seeing it sort of in low numbers across the field. Are you seeing hot spots where there's a lot in one corner but none in the rest of the field? That's important for the grower to know, sort of, relative risk, whether or not they can just do a spot spray on the outside of the field, on the field edges, or whether sort of different management is necessary. No real threshold, so that's why sort of just describing the distribution across the field is important. Thrips are another sort of mid to later season pest that you'll be looking for. They are quite small. They can move quite quickly. Also have a very large host range. Typically, you will see flushes of thrips after, for example, wheat is cut midsummer. There'll be lots of thrips in that wheat. Once it's cut, they'll flush out of that crop and into a nearby host crop. So 1 week you could have very little thrips, and the next week the entire one side of the field is covered in thrips. So very important to scout field edges with this pest. Really damaging later season to cabbage and cauliflower. Remember I mentioned at the beginning the development matters. Well, if you have sort of developed cabbage and cauliflower crops right when wheat is being cut, it's very critical you monitor any thrips moving out of that crop and into the cabbage or the cauliflower that's near harvest. The adults can fly. They're usually a little bit darker. You can see here they're winged, and the nymphs and the larval stages are usually a pale yellow. So they are quite quick. If you pull open the leaves and you see them, they will start to scurry away and to get out of the sun to hide again. So you kind of have to be quick when you're turning over the leaves and know what you're looking for. Why is it a problem? Well, their feeding damage, especially on cabbage, creates scar tissue. So it gives it sort of a gross, warty appearance. Kind of looks like edema, if you're familiar with that, which Travis may get into later on in the physiological problems. Cauliflower, you sort of get brown and tan streaking on the actual cauliflower head, so that marketable portion is also ruined. So I mentioned already, they're sort of like to hide out. So they'll sort of be on the outer leaves first and then move to the inside. They'll get between layers of the cabbage, especially, where you won't be able to see them. It'd be hard to identify, but they'll still be doing their damage and still be making that cabbage unmarketable. No real thresholds, but similar to aphids, describe the distribution. Are you seeing high numbers on the edges? Are you seeing sort of low numbers across the field? And relay that back to the grower. One thing to keep in mind with thrips is that they do have lots of natural enemies in the wild. So if you are seeing a lot of their natural enemies, that's also something to note, where a lot of times, sometimes if they're low numbers, they can actually keep the thrips populations in check. So minute pirate bugs, you can see here, are great predators of thrips. Very distinct, small insects with those distinct yellow to white patches on the back. So if you're seeing this minute pirate bug, that's actually a good thing. And if the thrips are in low numbers, you can report back to the grower that thrips are in low numbers, we're monitoring it, but we're also seeing a lot of natural enemies and beneficials that may be keeping the population in check. There we go. Tarnished plant bug. You can see here as an adult, a little bit larger than that minute pirate bug, does have sort of a similar look, as you can see on its back, but minute pirate bugs will be much smaller than tarnished plant bug. Where'd it go? There it goes. So quite small, and the tarnished plant bug will be a little bit larger, 6 to 7 millimeters, yellow triangular area, sort of at the top. Typically you'll see distinct here and at the sides. The nymphs are green and they have little spots. Sometimes they can be easily misidentified as aphids, but I'll show you that on the next slide, how to tell the difference. Very large host range. They also feed a lot on weeds and weedy areas. So sometimes they'll be on the edge of the field and move into the field when they're feeding on weeds, or in the other rows outside the field. The damage is similar to thrips damage, where you have sort of brown streaking on the curds and the actual marketable head. On broccoli, similar thing, dry, shriveled, sort of brown beads across the head as it feeds. And Brassica leafy veg, it's important because it can damage the flower stalk and cause flower problems as well to that marketable portion. There it goes. I mentioned that green nymph stage of the tarnished plant bug can often be misidentified as aphids. The aphids will have cornicles on the back, so they'll have little spikes coming out the back of them. Tarnished plant bug nymphs do not. They also have little spots on them. And the tarnished plant bug nymphs will be quite quick. They'll be running away, whereas aphids are a lot more slow and sluggish. If you turn the leaf over, this will run away and try and hide. The aphids will kind of just sit there. So that's how you sort of tell the difference. No established thresholds, similar to thrips and to aphids. Monitor the edges and report back to the grower the numbers and where you're seeing the damage. Flea beetles, and very important pests, especially on certain brassica crops, canola, if you're scouting any canola for sure, and also a problem early and later season in a lot of our brassica vegetable crops. We have two main species here in Ontario. The first one is a crucifer flea beetle. So that's sort of an all black flea beetle, and the striped flea beetle. So that one has two stripes on its back, very distinct. Attacks all brassicas, but prefers leafy, canola, mustard, and cruciferous weeds, actually. So it overwinters as adults, and sort of at the first warm period, it emerges. So if you have any seeded brassica crops, this can be a huge issue where that very weak and susceptible seedling emerges, and the same time as these flea beetle adults emerge, and they'll feed on that small amount of leaf tissue and basically kill the plant as well. You usually only see one generation. So when they emerge, they will feed, they'll lay their eggs, the new adults will emerge, and those adults that emerge in late July will feed, and those will be the ones that then overwinter into next year. So luckily there's not multiple peaks in generations, but the adults will emerge from the winter, they'll feed, lay their eggs, and those adults will feed a bit and then overwinter the next year. Prefer younger leaves and seedlings. They can tolerate one beetle per plant up to the sixth leaf stage. Later on, once the plant is established, flea beetle damage is much less important unless you are talking about leafy brassicas where you're actually selling the marketable leaves. Another problem with flea beetles is that they allow black rot to come in. So black rot is a bacterial infection that Travis will talk about in the pathology section. Very damaging, very critical brassica pathogen, and flea beetles leave entry wounds and holes for that pathogen to spread. So what are you looking for? In terms of damage, you're looking for shotgun holes. You may recognize this picture. Looks like somebody took a shotgun and just shot out the holes. A bunch of random scattered holes throughout the leaf. The holes will usually be ringed with brown or dead tissue. They are very active. They're called flea beetles for a reason because they act like fleas. Once you get near them, they will jump in the air and get out of the way. So it's important to be mindful whenever you're walking through a field that you're scouting, but especially in a brassica field, to know the spot you're going to and monitor as you come closer, because a lot of times the flea beetles will jump off before you get there. So you'll be doing your spot checks and looking underneath the leaves, but by that time the flea beetles already be jumped off. So make sure that you're looking for flea beetles while you're walking through the field and as you're approaching your spot checks. They like sunny areas and isolated plants. The threshold is the average number of beetles per plant. So that's why it's important, especially to count them as you approach the plant, because it's important for your threshold and to actually get an accurate count. Up into the sixth leaf stage, less than one per plant is sort of cause for concern. And then after the six leaf stage, as I mentioned, it's not really a huge problem unless you have a lot of black rot, or as I mentioned, it's a leafy brassica. There it goes. Some of the more, I would say, minor pests that you may see is as a leaf miner. There's several species in Ontario. They have quite a wide host range. So you may see serpentine, vegetable, or pea leaf miner. Quite small little insect, black and yellow. It lays its larvae inside where they feed basically in the leaf. You'll see these really distinct mining pathways and damage inside the leaves. Very distinguishable. And there's not really a developed threshold. If you see lots of damage, then report that back to the grower. In general, it's not a problem unless the population is very high. There it goes. Swede midge, one of the most important brassica pests if you are growing head brassicas such as cabbage, broccoli, or cauliflower. It is a tiny fly, very difficult to see, very difficult to monitor for. It overwinters as an adult, and it basically lays its eggs sort of at the growing point. These eggs hatch very quickly within 3 days, and they feed basically on the growing point in the meristem, destroy that growing point in meristem, cause a bunch of random growth. After 2 weeks, they will fall off the plant, pupate, and then either go dormant if it's later in the season or start another generation. There we go. What are you looking for? Well, as I mentioned, they're laying the eggs and doing the damage near the growing point and meristem. So that means you're looking for rotten, destroyed growing points and meristems, distorted leaves. Multiple heads on cabbage is usually a good sign of Swede midge, although it could also be other issues, but usually multiple heads, warped and weird damage at the growing point, and rotten, no heads, basically. The rot in the west tissue that you'll see is usually from secondary pathogens. Right, so at the growing point, it's like a cup. So there's a lot of water that usually sits in there. And if you have some damage caused by the Swede midge larvae, well, then it's an easy entry point for bacteria to get in there, and then soft rots take over. And that's why you usually see a lot of wet rot as well with this problem. To monitor for them, there are pheromone traps that you can use, and it's highly recommended, especially if you're growing a head brassica, that you do use pheromone traps. It's important to know the local population, because that really, they're very weak flyers. So your local population is really what determines your risk of damage. It's recommended you check the pheromone traps quite regularly, a couple of times a week if possible. And your thresholds are 5 to 10 males per trap per day for cabbage, and 1 to 5 males per trap per day for broccoli. So it's a lower threshold for broccoli. You can also scout for symptoms in the field. So those symptoms I mentioned, right? The growing point issues, distorted growth, rotten in the meristem area region. That's sort of what you're looking for. It's important to be able to identify males as well on those pheromone traps. So what you're looking for is sort of those spicules at the end of the abdomen, and also distinct head antennae. I don't know if it's antennae. I'm not an entomologist, so. So, oh boy, there it goes. You also want to look at brassica weeds within the crop and also on the edges of the field. These can also be hosts and reservoirs for these insect pests. So, your shepherd's purse, your dog mustard, your stinkweed, they're all brassicas and in the mustard family. So a lot of these insects that I've just talked about will also infect and feed on these crops. So be aware of your weeds and be mindful and keep an eye out for them as well. Okay, I didn't see any questions pop up. If you do have questions, feel free. And if not, I ask you to open up your Slido again on your phone. And we're going to go through those questions that we went through at the beginning and see, hopefully, if we score a little bit better than we did at the beginning. So take out Slido on your phone, type in Brassica, and you should see the first question there. What is this insect? Yikes. Hold on one sec. There we go. So hopefully you're all able to find it back. Slido.com, type in Brassica. You should see that first question. What is this insect? So you can see here, green caterpillar. Give up remote control. There we go. I won't say any more because I don't want to give it away.
All right, I think we've got everyone. The correct answer is cabbage looper. Nice, well done.
100%, well done.
Wow.
Moving on, next question. What caused this feeding damage? So very distinct. I also don't want to say too much and give it away. But feeding damage there on that leaf. Is it cabbage looper, tarnished plant bug, cabbage maggot, flea beetle, diamondback moth, or seed corn maggot? Looks like we're all in.
Yep.
81% flea beetle. Correct answer. Nice work. Okay, question 3. What is this insect? Is it cabbage looper, B, tarnished plant bug, C, cabbage maggot, D, swede midge, E, diamondback moth, or F, seed corn maggot? Couple more seconds. If you don't know, you can just guess. Hopefully you do. And it looks like you do. Cabbage maggot 88%, seed corn maggot 12%. So we would accept both answers because, as I mentioned, it's difficult to tell the difference between the two unless you have a dissecting scope and you're an entomologist. So in this case, it was likely cabbage maggot, but we'd accept both cabbage or seed corn maggot. What is this insect? Specifically, what is that insect egg? Is it cabbage looper, imported cabbage worm, cabbage maggot, swede midge, cabbage looper, or seed corn maggot?
Aster yellows, downy mildew, or black midrib. 5, 4, 3, 2, 1. What's wrong with this leaf? So we have a lesion with a few rings inside. Is it alternaria, botrytis, bacterial leaf spot, downy mildew, soft rot, or black rot? Five, 4, 3, 2, 1. And our last question, what is causing this symptom development on this cabbage plant? Alternaria, botrytis, bacterial leaf rot, downy mildew, fusarium yellows, or aster yellows? Five, 4, 3, 2, 1. All right, we'll get on to talking about disorders and pathogens. So it's good to know the definition and the difference between a symptom and a sign. A symptom is a plant's response to stress. So let's say that you have a cabbage plant that's turning yellow. There could be many things that could be responsible for that yellowing on that cabbage plant. It could be a lack of a nutrient like nitrogen. It could be phytoplasma like aster yellows. It could be a fungi like fusarium yellows. All of these are different forms of symptoms, stresses that are being caused on that cabbage plant. Whereas a sign is something where we are able to confirm for sure, based on physical evidence, that a pathogen is responsible for what we're seeing. So a sign could be fruiting bodies that is very specific to a known pathogen of brassicas. It could be specific mildew, where you're able to see the branching of the mycelium if you were to put that on a slide in a microscope. It could be bacterial ooze if you cut a stem open and you see the bacteria streaming from the stem. In insects, it could be actually seeing the specific insect or a specific gall or frass that that insect is leaving. And it's also good to know the difference between biotic and abiotic. So biotic would be our living pests and pathogens, whether that's insects or a fungi. And then there are abiotic factors where these are non-living things causing a stress resulting in a symptom. And in many cases, there is often not a defined line between healthy and affected tissue. So working with disorders and looking for or confirming something is caused by an abiotic stress is not easy. And the best thing that you can do is look for patterns across the field and in the specific plant. So what parts of the plant are infected? Look at the roots. How are the symptoms progressing from early to late stages? Is it all of the current new growth, regardless of plant stage, that's affected? Or is it only the new growth after a certain stage in the crop? Make sure to check the lower and upper sides of the leaves. Is the damage only on one side of the plant versus all of the plant? Are there differences between cultivars? Are there differences in low spots or in high knolls where there might not be as many nutrients and potentially a nutrient disorder in those higher areas of the field? Are there differences between planting dates? Are things scattered at random throughout the field, or is it are the symptoms only visible around the field edges? So figuring out what is causing the symptoms is not easy, and that's why looking for patterns is key. Many abiotic disorders are caused by imbalances or deficiencies in nutrients, and that's why it's always good to have a soil or a plant nutrient tissue test conducted, and to do that yearly. So now we're going to get into some abiotic disorders that have been observed on brassicas. So on the left, we have Brussels sprouts showing leaf scald. And this is, weather maybe over 30 degrees Celsius, 90 degrees Fahrenheit. That's just too hot for the brassicas, mixed with maybe a little bit of irrigation water and some direct sun. You get those cells exploding, and the resulting is this wilt and these white lesions developing on the leaf. And then there are other abiotic disorders that could be genetic, like chimera, which is kind of like variegation, but the lack of pigment developing in the cells. And some plants are genetically susceptible or more likely to show chimeric symptoms than other cultivars. With the cold nights we've been experiencing in the last couple weeks, there's a good real chance that we will have seen some sort of frost injury on any transplanted brassicas that were planted early. And so look for curled leaf edges, blotches or spots similar to that of heat, and maybe, if it got real cold, you have that bursting of the inner cells of the leaf, and with those cells gone, you get blotches forming. So depending on the temperature, the cultivar, the field, and even differences in low spots versus high spots of the field, whether there was wind, whether you irrigated the day before, the symptoms are quite different. Leaves might turn white and just drop off. You might get lower, larger, older leaves turn purple and then wilt. Red heart is an irreversible internal leaf tissue damage where those cells have frozen in, or damage inside the leaf, and the result is that leaf is turning more purpley. Later in the season, you may see symptoms that are caused by too much moisture, and you might have a dry spell and 2 inches of irrigation water might have been irrigated the night before a thunderstorm where we got a downpour, and that resulted in another couple inches, and too much moisture, and the roots are waterlogged. They're unable to take up oxygen, and the leaves might wilt, turn yellow. In broccoli and cauliflower, your florets might bolt, and even cause an uneven curd development. In cabbage, you might even see that some cultivars take up nutrient or water too quickly and will split open and crack. Where there is hot, drying winds, you might see tip burn developing on brassica leaves as they lose water, and through the leaf tips where there are hydrothodes, this cell that allows for water to escape at night when the water pressure is high, and take in that water droplet in the early morning as the relative humidity goes down. And those hydrothodes are a easy area for that brassica plant to lose water. And then if it's unable to get water back when it needs it, those tissues don't have enough water, they end up dying. And the result is in cabbage, as that head is forming, those dead, dry tissues are trapped inside the cabbage head, leading to internal tip burn. Besides chimera, there are other genetic abnormalities that may affect the plant. And they could be cultivar dependent. So some cultivars are more susceptible to having blind plants. And this is where the plant just fails to grow any more leaves, and that plant is now referred to as blind. It's not growing any more new leaves. The leaves that are existing might get larger, but that's all the leaves the plant's really going to develop that year. And as a result, you're not going to get a head or whatever the favorable trait of that brassica is. The damaged growing point that could result in a blind plant could also be from mechanical damage, or it could be insect damage like sweet midge, as Dennis referred to earlier. In the past years, we've seen hollow stem in both broccoli and cauliflower, and this is extremely cultivar dependent. And if the cracking's internal, it generally does not cause any problems because no bacteria, no fungi are getting in there and colonizing this tissue. It's inside the plant. It's not damaging the plant in any way. In some cultivars, this has been traced to boron deficiency, and boron required for the movement of carbohydrates. And if there's not enough boron present, it results in the cracking of the stems. On cabbage, you might see black speck, and you know the cabbage might go into storage and look just fine. And over time, there might have been a couple drops of that head, or it got squished against the sides of a crate. And over time, these bruises appear on the outer leaves, and they can be in the form of black speck. So it tends to be localized in various leaf parts, and it typically is only shown after a bit of storage time or time in storage. Next, we have black midrib or black petiole. And this is a cabbage disorder that is thought to be an imbalance of phosphorus and potassium levels in the field. So if this is the case, increase the potassium in the soil without increasing the level of phosphorus. And that's easier said than done. And we think this is likely linked to high rates of nitrogen, and it is cultivar specific, and it also can be affected by the storage conditions that the cabbage is being stored in. Splitting of cabbage, I mentioned that it could be from too much water, but it's typically due to too much nitrogen. Lots of growth, susceptible cultivar just can't control, and this overgrowth causes the cabbage to split open, and it just can't keep up. There is brown beating that can occur in broccoli, and this is where the buds or little florets turn tan and brown. And we often see this in periods of rapid growth, high temperature, and abundant moisture. Could also be because of a fungal pathogen like Alternaria that we'll talk about in a bit, and maybe even tarnished plant bug feeding on the broccoli floret. So if you happen to see tarnished plant bug in the field and you come back a couple of days later and you see brown bead on the broccoli, there's a chance that those two things are linked. And then there are head disorders where specific cultivars may have lose their crown, lose their shape due to a lack of nitrogen. So Captain and Decathlon, with adequate amounts of nitrogen, they form a nice head, but with low amounts of nitrogen, they become uneven and are not a nice cup. In cauliflower, you can have the heads appear fuzzy in appearance, and this is known as riciness. So cool and then hot weather at various stages of floral initiation can cause this to happen. Talked a bit about nitrogen deficiency being the cause of splitting in cabbage. And this is where the lower leaves turn bright orange, red. It sometimes looks like fusarium yellows or in some cases aster yellows, which we'll be discussing shortly. With potassium, instead of the whole leaf turning yellow, you often see the discoloration around the leaf edges or giving the symptoms of leaf scorch. And other stresses like frost and rots may predispose the plants to a potassium deficiency. Manganese deficiency is not often seen in Ontario. Older leaves become pale yellow, marbling between the veins. And with molybdenum deficiency, you get whiptail plants being created. Long, narrow leaves form and lots of leaves. So molybdenum is required for chloroplast maintenance and nitrate reduction. And without it, you get these smaller, thin leaves. Does not occur often. Other damage that's not abiotic may look like a blind plant, but it could be sweet midge. So if you do happen to see distorted leaves in that meristem, pull back those small leaves, get in there. If it's rotted, you could pull out that rot and you might be able to see the remnants of sweet midge there. So now we're going to get into the major pathogens of brassicas. We'll start with ones early in the season, like damping off. There are two types of damping off. There is pre-emergence and post-emergence. So pre-emergence is when the fungi attacks the sprout after it's the seed has germinated, and before it reaches the soil surface, the pathogen overtakes the plant and kills it, and that is our pre-emergent. Post-emergent is where that sprout makes it above the soil line, and then we start to see the damage to the above ground parts of the plant, whether that's wilt or other symptoms we'll discuss shortly. So this is an issue because if you're just planting the seeds in the greenhouse and you think that the viability should be 90 or 95%, but it's actually 60, well, dig into that plug and see if the seed is actually germinating. Is it germinating, and is that shoot and root turning brown? And that there's a good chance that it could be Pythium, Rhizoctonia, or even Fusarium causing pre-emergent damping off. So what can we do to prevent damping off in the greenhouse and early in the field is to start with sterilized potting media. Avoid seeding in cool plotting soil. So if you're bringing it in from the barn to your transplant greenhouse, let it sit there overnight and warm up. Don't use it right away. Avoid overwatering. waterlogged soils are not good. And we're moving towards more plants per tray, and we're reducing our cell size, and that's causing potentially an overcrowding of seedlings. There's not as much airflow between the plants. You're creating a very dense crop canopy. Without enough light, you might get leggy plants. And if you apply too much fertilizer at seeding, it could predispose the plant to damping off. So what can we do? We improve water management, we ventilate, we improve the lighting, and we could apply a preventative product like Root Shield or Serenade Soil, where there's a biological that can colonize the newly germinated roots and compete or antagonize that Pythium or Fusarium and prevent it from colonizing that root, or use a seed treatment like Thiram so that that seed can break through the soil surface, get established, and have a good chance of surviving. Wire stem is a form of Rhizoctonia, and you saw Rhizoctonia in the damping off section. Later plantings, or when right after you transplant in the field, wire stem could become more apparent. So the symptoms are very similar to Pythium or Fusarium. It's basically a canker at the soil surface that girdles the stem, prevents the stem from growing any larger, whereas the I guess the stem above that crown and the roots are still able to grow, giving it this wire-like appearance. So you're looking for that wire close to the soil surface. You're looking for lesions where that stem is attached to that crown. And this could also be confused with wireworm damage in the field. So what can we do? Start with clean soil. Oh, I'm going one ahead here. Life cycle. So basically the infection occurs when the plants are seeded and there was improperly cleaned trays or equipment or benches, and Rhizoctonia is spread by wind, water, or even equipment, and it makes it to that growing plant, colonizes the crown, and as it colonizes, it creates that wire-like appearance. So what can we do? Make sure that those propagation trays and equipment are clean. We can use Thiram or fungicide treated seed or a biological that colonizes the plant and prevents the Rhizoctonia from getting established. We can avoid overcrowding in the greenhouse, improve ventilation, avoid planting in soil temperatures that are below 10 degrees Celsius or 50 degrees Fahrenheit. Avoid banding fertilizer close to the seed. Maybe cut back on your fertilizer, and instead of a weekly application, do a lower concentration daily. Implement a minimum of 3-year crop rotation when you're transplanting into the field. If there's Rhizoctonia present in the field, it can colonize those transplants quite quickly and then lead to the symptom development we saw in the previous slides. Our next pathogen is blackleg, and this is more of a pathogen we see in canola and not a pathogen that I have seen in horticultural grown brassicas. So just like damping off or Rhizoctonia wire stem, the fungus is attacking the roots and the crown and causes cankers at the crown. So the plants are stunted. You get some girdling of that stem, and if you were to zoom out and take a look at the leaf, there's a good chance that you'll see these lesions with a whole bunch of peppery spots on them. And these peppery black spots are known as pycnidia, and they can be seen mainly on the leaves, but also the stem. This particular pathogen is a cool season pathogen, 5 to 10 degrees Celsius. So early in the field, you might see it. If you cut open that stem of the Brassica plant, you could see these black lesions inside that cross section, causing problems to the vascular tissue. So here's a better picture of that pycnidia and those spores on the leaf. What can you do? Treat the seeds, implement a 4-year crop rotation, remove susceptible Brassica weed hosts during the rotation, and use well-drained fields during months that are typically wet and cool. The next fungi we're covering is Alternaria, and this is our main foliar pathogen of Brassicas. So we can distinguish Alternaria by these circles, these concentric rings on the leaf. It generally starts on the older leaves first, so this is why it's really important when you're scouting to check those lower leaves. And if spots start to colonize and cover more than half of that leaf, you can expect those leaves to fall off. And then those fallen leaves are now inoculum for other plants in the field or other blocks down the road. So it is important that after you harvest this particular block, that you incorporate the residue and get it to break down as quickly as possible, thereby breaking down those Alternaria spores as well. In broccoli and cauliflower, you get the black streaking of the curds if you were to do a cross section of that floret. In Brussels sprouts, you get spots on the actual sprouts, and this pathogen is also called black spot. So what happens? These spores are spread by wind, rain, equipment, and people. If the weather is above 15 degrees Celsius for 9 hours, those spores are able to germinate and infect the crop. And I mentioned earlier, these leaves fall, the fungi grow on the leaf and are able to infect the marketable crop that surrounds those leaves. So there are seed treatments that can be used. You want to space plants far enough apart so that there's adequate ventilation, and that will greatly help with larger plants like Brussels sprouts. Rotate with cereals or corn and control chewing insects. In the field, you want to avoid overhead irrigation during head development. If there's a chance to go on drip in fields that are really an issue with Alternaria, it might be worth giving that a shot. And I mentioned earlier, you want to incorporate those debris as soon as you can, get rid of those spores and make it harder for them to infect later seedings. Sometimes we see Alternaria develop in storage, and that requires the plants to be protected by a fungicide before they're harvested. So stable temperatures in storage below 4 degrees Celsius inhibit the growth of fungi, but if your storage is above that temperature or it takes a little while for it to get to storage, you could still see these fungi develop. As a scout, you want to inspect 10 older and newer leaves of each scouting stop, record the average number of lesions per leaf, and note any spotting on florets or heads when inspecting close to mature plants. Our next pathogen is downy mildew, and In seedlings, we'll see yellowing of cotyledons and this white cottony mycelial growth on the underside of the leaves. In bad infestations, in ideal conditions, those leaves turn dry, papery, and they stay on the plant. They rarely drop. And so if you were to pick one of these leaves off the plant and hold it up to the light, you should be able to see that white mycelial growth on the underside of the leaves. Cauliflower, you get dark gray spots appearing on the curd, and streaking can be found in the florets, similar to that of alternaria. And in broccoli, we see no spots on the beads, but the gray streaking occurs beneath the beads. So this pathogen overwinters in roots and in crop debris. So this is why it's really important to incorporate last year's crop as soon as it's harvested. And the fungus develops when there are 3 hours of cool temperatures between 10 and 15 degrees Celsius with a heavy dew or a fog, anything that, might be 80% relative humidity and higher. And then you see those symptoms in as few as 4 days after that has occurred. So what can you do? You can plant resistant cultivars or plant into well-drained soil, avoid overhead irrigation in the evening, irrigate in the early morning, and allow that crop canopy to dry out and limit the amount of hours that that crop canopy is actually wet. There are preventative fungicides registered. I keep on mentioning incorporate harvested tissue into the soil, allow those spores to break down and not infect other seeding or blocks. And as a scout, also inspect those 10 older and newer leaves at each scouting stop, record the percentage of leaves and the number of lesions per leaf. So far, we have discussed fungi or oomycete pathogens that are able to be controlled by fungicides. So now we're going to jump into some bacterias and virus. And the first is turnip mosaic virus, which causes wilting or wrinkling of the leaf tissue, similar to frost or herbicide damage that we could expect in some cultivars. So cabbage head may show symptoms several layers deep if you're peeling back those leaves and after 2 to 5 months in storage. This is going to be sporadic through the crop. It's going to be from those aphids feeding on one plant and then going to another. So it's generally not going to infect the crop uniformly. So if you happen to find 1 or 2 cabbages in storage that show this symptom development, and the rest of the cabbages are fine, you might be able to rule out abiotic injuries and instead it being caused by a virus. So this virus is transmitted by aphids, and this virus overwinters in volunteer rutabagas, winter canola, and other brassica weeds. These aphids go visit those plants, pick it up, and then carry it to the brassica field. So what can we do? I'd rogue out plants if I found them in the field throughout the season. I document the location just to see if I start to see patterns. As a grower, you can avoid planting near fields that were planted with rutabaga or canola in the previous year. Get rid of volunteer rutabagas and manage aphid populations to reduce the rate of virus spread. Moving on to bacterias targeting brassicas, we have a few. Pseudomonas causes these small circular lesions in the leaf with these light green halos surrounding those lesions. So these spots dry out, and as those spots dry out, those cells will disappear from the leaf, and you'll often be what you'll often be left with what looks like insect damage, bullet holes in that leaf. Um, so it could be mistaken for very precise flea beetle damage. It overwinters in crop debris and on seeds, and these bacteria cells are spread by splashing of rain, irrigation water, as well as insects. So what can we do? Implement a 3-year crop rotation, obtain seed that is tested to be free of Pseudomonas, avoid overhead irrigation or irrigate in the early mornings and allow the crop to dry out. Um, ensure that our irrigation water is not contaminated with Pseudomonas, and manage insect pests that are able to vector the Pseudomonas bacteria. Bacterial soft rot is something we tend to see later in the season, especially as the temperatures increase. And the symptoms are a result of many different bacteria species, such as Pectobacterium, Erwinia, or Pseudomonas. And you're getting this moist, mushy appearance sometimes on the inner leaves, sometimes on the outer leaves. In broccoli, you get that uneven head formation, and that uneven head allows for water droplets to collect on that broccoli head. Bacteria cells could land in those water droplets, and if that broccoli head was wounded by an insect or even probed by an alternaria fungal spore earlier in the season, these bacterial soft rots are able to get into that wound site, colonize the plant, and see the symptoms here. If you're brave enough to smell it, you're not going to like that smell, and that's pretty characteristic of bacterial soft rot. So these bacterial cells overwinter on infected tissue. They can overwinter within cabbage maggot and also equipment and containers. They're spread by direct contact by soil water, the splashing of rain or irrigation, as well as sweet midge, cabbage maggot, and chewing insects like flea beetle or tarnished plant bug. What can we do? Practice a 3-year crop rotation, test the seed before planting, avoid overhead irrigation or prolonged hours of having that leaf or being wet, and manage our insect pests. And the next bacteria is deadly, and it needs to be caught as quickly as possible once we see it in the field. So black rot is called by Xanthomonas campestris, and these bacteria leaves are, so let's say that the bacteria are in the field, and these plants overnight start to have water droplets that form on the tips of the leaf by those hydrothodes. So at the end of the veins of the leaf, we can find hydrothode cells, and you'll see water droplets collecting there. If a Xanthomonas cell is able to land on that hydrothode, on that water droplet, when the relative humidity goes down in the early morning and late morning, and the plant needs to pull in moisture, it will actually suck in that water droplet and pull that bacteria cell inside the leaf. That leaf, the phloem, the sugars that are inside the leaf is a great environment for those black rot, those Xanthomonas cells to divide, replicate, and over time, they'll actually clog up the veins of that leaf. And it will grow closer and closer to the stem and to the root of the plant. So as those vascular tissues become clogged, all of the leaf tissue that was serviced by that vascular tissue is now not getting enough nutrients. it turns yellow and starts to die. And if you were to pick this leaf and hold it up to the sun, these veins would be black while these veins would be clear. So 1 in 10,000 plants results in a field epidemic, and late infections can provide wounds for other bacterial rots to get in. And these bacteria stick around. So you want to deal with this as soon as you find it, not leave it for later. And you might be, shooting yourself in the foot for future plantings of brassicas. So if you were to cut open that leaf or the stem in this cross section of Brussels sprouts, you'll actually see those black spots. That's the xylem, or the vascular tissue that is clogged with Xanthomonas cells. And in Brussels sprouts, it can create these lesions on the sprout. And this is characteristic what we're going to see on all of our brassica leaves, this V-shaped lesion. So it starts at the tip and works its way back. And since it's kind of contained, or the tissue that's dying between the veins, those veins are creating V's, and that's why you get those V-shaped lesions. So implement a 3-year crop rotation. Make sure that you're starting with good seed. Disinfect greenhouse surfaces if this has been a problem in the past. Work crop debris into the soil as soon as possible. Control all brassica weed species, because that is one way that Xanthomonas can overwinter, even if you haven't grown brassicas in the last few years. This pathogen can spread quickly, so it's best to avoid it altogether. Generally, if it's found, rogue out those infected plants if that's possible. Quava is registered for suppression. And as a scout, you want to catch this early, record the percentage of leaves infected, the average number of leaves or lesions that you find on the leaf. Hold those leaves up to the light and observe the black veins. We're going to jump back to fungi for a few slides and start with Sclerotinia. Sclerotinia fungi causes this white mycelial growth and produces these small black sclerotia that look like mouse droppings. You saw that picture in the quiz with all those black spots. Those are sclerotia caused by Sclerotinia. It's also called white rot, and as the mycelia colonize the tissue, the leaves wilt and can cause rot. So these sclerotia, these little black sort of mouse droppings, they drop to the soil once they've been formed, and can stay in the soil and remain viable for up to 8 years. And so when these spores reach the top inch of soil and the soil remains wet, and there are 10 days where it's over 10 degrees Celsius or 50 degrees Fahrenheit, these spores are able to infect, colonize the stem or the leaves, and move on to the rest of the plant. In general, Sclerotinia is a weak pathogen on brassicas and gains access through plants in wounds or damaged areas from insect feeding or other pathogens. So there are a bunch of management strategies available, and as a scout, you would count out 25 plants and record the number of plants that were infected with sclerotia. Our next fungal pathogen is Fusarium yellows, and it's caused by a specific subspecies of Fusarium, oxysporin, and basically it creates symptoms that could be mistaken for black rot. You'll see black veins in the leaves and yellowing of the lower leaves, not the top leaves that you could expect in a nitrogen deficiency or aster yellows. So Fusarium yellows is very characteristic of the lower leaves. So if you cut the crown lengthwise at soil level, you should see a black stain of that vascular tissue, and that is very characteristic of Fusarium yellows. This pathogen is soil-borne, and the spores enter the root, colonize the plant, reduce the nutrient uptake of the roots, and cause the older leaves to yellow and die first. And as that fungi is replicating inside the plant, it is releasing a toxin that causes the plant to wilt and eventually kills the plant. And these specific races of Fusarium are very host specific, and the cabbage race may not infect radish. So as a scout, I would look and make sure that you don't see any V-shaped lesions and rule out that it's not the Xanthomonas black rot, as we saw earlier. You want to cut open the stem, and if you see the discolored xylem ring in the stem, that is pretty characteristic of Fusarium yellows. So Fusarium yellows starts from the roots and moves up, while black rot starts from the leaf tips and moves down. The next few pathogens attack the roots, and the first is sugar beet cyst nematode. Look for patches in the field of wilted or stunted plants. And what happens is the nematode hatches from an egg, feeds on the root, and the females attach to the root and swell up into this white cyst that is just full of eggs. They can survive in the soil for up to 12 years, and this whole life cycle can be completed in as little as 30 days in favorable soil conditions. So the best thing that you can do as a grower is crop rotation. Plant non-hosts in the off-brassica years, such as grains, corn, soybeans, and alfalfa. As a scout, do not pull up plants that are stunted by hand. The roots are weak, they will break off, and you will never see these white or brown cysts that will allow you to identify this pathogen. This is why everyone needs to have a trowel and dig up that plant and gently knock the soil off the roots. The next nematode is root lesion nematode. And the symptoms of root lesion nematode can look like nutrient deficiencies or wilts. The plants appear stunted. It can be seen in patches, or it can be seen as rows, as you know, the cultivator is moving soil in a linear fashion sometimes through the field, and that can result in rows of plants showing root lesion nematode damage. So this nematode penetrates and feeds inside the root. The females lay eggs inside, and the life cycle of this nematode can be completed in as little as 40 days. So if you determine that this is the nematode of concern, you want to plant early and avoid rotations where there is highly susceptible crops. Don't plant brassicas on brassicas on brassicas. And remember to have that trowel handy to dig up plants and inspect the roots. Our last nematode is root knot nematode, which also causes stunting, wilting, and you could see yellowing of the leaves. It begins as small patches in the field, and in severe cases, it can be acres of the field. Same issue as before. These knots weaken the roots, and without a trowel, you're leaving that symptom inside or below the soil line. So get out your trowel, dig up the roots, look for these small root galls that look like nitrogen fixing nodules that you might see on legumes or soybeans. So one female root knot nematode can lay 1,000 eggs within its life cycle. And if favorable conditions are met, it can complete its life cycle in as little as 25 days. And so you can imagine that these numbers multiply and grow quite quickly. So identify the nematode, and if it's root knot, avoid susceptible crops like onions or barley. Observe a minimum of 3-year crop rotation and make sure that you control your susceptible weeds. And as a scout, you're going to use that trowel, carefully dig up roots and look for galls on the roots. And the last pathogen we're discussing today is clubroot, which is also a soil-borne pathogen like the nematodes, and it causes clubbing of the roots. And because those roots are clubbed, you get that wilting that is similar to the nematodes. You might get yellowing of the foliage, and on hot days, it's very characteristic of the plant to start wilting. And so 1 gram of soil can contain 100 million clubroot spores. And these spores can last in the soil for over 23 years. So the galls that you see on this slide could be caused by as few as 1,000 spores per gram. So once this pathogen is in the soil, it is one of the hardest to control. The best thing that you can do as a grower and as a scout is to prevent that pathogen from ever reaching the field in the first place. And that's why biosecurity is so important. Make sure that equipment is clean when it's coming from another field. Make sure that there's no soil, no dirt remaining on your shoes, your boots, on the tires of your vehicles or of machinery. No chemicals or fumigants have found to suppress spore loads to a level of suppression or control that is noticeable. You only need 1,000 spores per gram to cause a severe infection. And if you're starting with 100 million spores per gram, even if you control 99.9% of those spores, you're still left with 10,000 spores or something, still enough to cause a severe symptom in the plants. So as a scout, use that trowel, pull out that root, look for those clubs. In general, if you pull out the plant without a trowel and you see clubs, it's most likely clubroot. But if you, whereas you will need a trowel to dig out the roots to see the knots or the cysts that we saw in the nematodes. So that is a summary of the main pathogens affecting brassica crops in Ontario. Our retired colleague, Mike Cialetti, always showed resources that were available at the end of the IPM presentation he gave. So I'd also like to highlight that these are great books to have on the dash of your truck or folded and kept in your back pocket and have handy when you're out scouting a field. I'd also like to point out that we have a Brassica section on crop IPM, which is also a great resource, but digital. And the slides presented today have had many contributors over the past years, and I would like to thank all those that are shown here. And before you go, I would like to go over those 5 questions in the quiz to show that you actually learned something with today's workshop. So our first question, what caused this damage? Botrytis, sclerotinia, rhizoctonia, aspergillus, downy mildew, or black midrib? got 7 votes in. It's really important now that you vote and that you get it right, because then it shows that you actually learned something. 5, 4, 3, 2, 1. The correct answer here is sclerotinia. We've got those sclerotia, those black mouse droppings on that cabbage head, and white mycelial growth growing over that cabbage. What caused this damage? Is it alternaria, botrytis, bacterial leaf spot, downy mildew, soft rot, or black rot? Five, 4, 3, 2, get your answers in. And our answer, everyone got right, was black rot. Right on. Our next question, what caused this damage? Botrytis, sclerotinia, rhizoctonia, aster yellows, downy mildew, or black midrib? Five, 4, 3, 2, 1.